Thanks for tuning in, everybody. My name's Stephen Knowles. I'm talking to you from Brisbane. I established the Alpha-1 Association of Australia in 2005. This is our second webinar aiming to keep Australians informed of all aspects of Alpha-1 antitrypsin deficiency. The webinars are being arranged by the Alpha-1 Association of Australia in collaboration with Alpha-1 Global. Our second webinar about genetic testing for Alpha-1 in the laboratory will be presented by Dr. Val Hyland of Pathology Queensland in Brisbane. Val has been a geneticist for 35 plus years. So over to Val.
Thank you, Steve. And thank you, Kevin. So I'm going to share my screen. Okay, so I'm going to talk about AAT or SEO PIN A1 gene testing. So I've got some references to open up first, and these are some references that I find very useful. The first one is called Genetic Home Reference, and the second one is Gene Reviews, which is perhaps something a bit more technical. And I'll just go to those. Okay, so I'm going to, when I type into this Genetic Home Reference in this search button, I put in the name of the gene, or I can put in AAT, and I bring up these two filters, and I'm going to open the first one. And I probably don't tell a lot of the audience about this gene and what happens when this gene is not functioning. But we've got quite a nice description there that this SEO PIN A1 gene provides instructions for making a protein called Alpha-1 antitrypsin. And this is a type of serine protease inhibitor. And this helps control several types of chemical reactions by blocking or inhibiting the activity of certain enzymes. And this alpha-1 trypsin prevents the digestive enzyme trypsin from breaking down proteins until trypsin reaches the intestine. And the alpha-1 trypsin also inhibits other enzymes, including a powerful enzyme called neutrophil elastase that is released from white blood cells to fight infections. And the alpha-1 trypsin protects the lungs from this neutrophil elastase, which can damage the lung tissue if it's not controlled. And the alpha-1 trypsin is produced in the liver and then transported to the lungs via the blood. So there are quite some interesting fields you can open up if you go back to this website later. I'm just going to click on this additional information and resources, and this button here it opens up. This is another Gene Reviews, which is, to some extent very, very technical, but really has plenty of information and is, very well written. And you can see from the top here, the last time it was revised was early last year. So just to give you an idea of the kind of testing that we do in the lab, I pulled up these summaries, and from the left-hand side, it's got testing that we've done in this year from July, which is really the end of last year, from the 1st of July to the end of December. And you can see that we've done slightly over 1,200 genetic tests with something called a TACMan. Last year we had over 25,000 tests, So we're kind of keeping on par, similar kind of numbers. And the previous financial year, we had done about 2,150. So this amounts to doing about 40 tests per week. Besides doing this, what we call a TACMAN analysis, and I'm going to show you a slide explaining that, we also do some DNA sequencing of the gene, but we don't do very many of those. There's on average about one a month. And usually the reason we would do that is because we haven't found any change in the gene by doing this TACMAN test, and we would then go and do full sequencing of the gene. Now, just go page down. I'm going to show you this assay, and I'm going to share another screen. Okay, this is I've prepared this slide for something else a few years ago, but I think it is quite a nice explanation. So if you imagine your DNA, and your DNA is double-stranded, and the first thing you have to do when you're beginning to analyze this DNA is separate the two strands. And it's quite simple to do this. We really heat the DNA up, and what happens is the DNA becomes single-stranded, so there is a free DNA strand. And then we have a special kind of probe that we put in, and this probe can bind to DNA, but it will only bind to DNA where there is a change in that DNA, and we call that DNA, that change, a variant. And this DNA strand that we make has what's called a reporter and a quencher, and they have special functions which you'll see. So when we hit the reporter with wavelength of a particular strength, it emits a signal, but this signal is captured by the quencher molecule, and we cannot measure it with another device. But in this polymerase chain reaction, here's what happens. We've got two other DNA fragments. These hybridize to the patient's DNA, and then they begin to copy the DNA using an enzyme called polymerase. So here's the polymerase binding to the double-stranded DNA. Here it is beginning to make DNA, and it just begins then to break that first probe that had bound to the DNA. And you can see the light is exciting this reporter, and it's emitting light at a different wavelength. But this quencher is no longer beside it. And the reporter is free in solution, and we can start measuring this activity. And this can tell us whether there is a variant or a particular kind of variant present in the DNA. Okay, I'm going to try go back to my share. Usually what we're trying to do with this TACMAN analysis is measure two variants, and you've probably heard these variants. They're called S and Z. And I've just brought some results from the last few years about these. So often before we test the DNA, some kind of AAT measurement has been done. And up in the top right, you can see some values there. So what were the different patient samples that had an AAT above 0.9 grams per liter, below that or just at it. So there's slightly less than 10% of them have a value of AAT which is less than 0.9. But you see the numbers here. What kind of numbers have 2 Z variants? And here in this year there were 6. What individuals had an S or a Z? And here there are 13. And I've given the values of the mean AAT protein in the serum in grams per liter. And you can see there that the ZZ individual has a lower value than the SZ. And then there are some other combinations there that have other values of mean AAT. So that's 1 year. This is the next year. And again, you see there are a small number of individuals. 20 had an S and a Z. 15 had a ZZ. You can see also in this slide, how long did it take us to actually do that analysis. So in this year, it's taking 9 days plus or minus 2 or 3 days. We've got the results in 2015, and then there are about 2,000 samples here, and we've got more individuals that we found with ZZ or SZ. This is 2016, and here I'm just measuring the level of AAT in grams per liter, really just for these individuals that I've marked in red. But again, you can see the ZZ values are pretty low AAT measurement, and the SZs are a little bit higher. 2017 results there. And we're beginning to just focus on looking at the S and Z, and we've stopped looking for what are some other variants. And our turnaround time has blown out. It's taken twice as long. And then our results for the end of last year, the last 6 months. Okay, so I'm going to go to these websites here and tell you something more about the gene. This is a website, it's called NCBI. I've put in the gene symbol, and it's come up with the various information that's available, and we tend to look at this reference sequence. Doesn't seem to be going. It's perhaps a bit slow moving, but I'll move to a next screen, and this probably shows the gene a little better. This is a European website called Ensemble, and if we look at the top on the right-hand side, this is first we call exon. It's not actually coding for any protein. The second, the third, the fourth, and the fifth are making DNA that will make messenger RNA, that will make protein. And you can see a lot of color on this slide, which is really all the variation that has been found in this gene. This is another website called Plenvar, and this shows 175 different variants, and I'm going to come back to this later. I was asked what kind of other variants do we find, and there is a very important variant called a null variant, and these null variants usually make no protein at all. And these are some of the null variants that we found in the lab. We've got 1, 2, 3, 4 here, and usually they go by a name. So someone has found them in a patient where they work, and perhaps give it a name associated with that. These variants are normally very rare. So the first variant we found was just on its own. It didn't have an S or a Z variant, and it had a value here of 0.69 grams of AAT per liter, which is something very similar to what you would see for an SZ individual. The second variant here was found in association with a Z variant, so it's pretty much like a ZZ, but instead of being ZZ, it's got Z plus another DNA change. And this value here of AAT in grams per liter is very similar to what you would see in a ZZ individual. On this slide, I've got what are called some of the missense changes. So down towards the bottom, we've got what is the change at the nucleotide level and at the protein level for the S and the Z. But we've got some other variants that we have found. But I think if you look at the AAT here in grams per liter, it's the one I've underlined here at 0.34, which is important for the patient. And then this fourth individual, which has a Z variant with something called a Bristol variant. And then sometimes we're finding variants which haven't been described anywhere before. They're invariably these, what we call missense. So instead of coding for a particular amino acid, they may code for another. This third one here is an individual which has an S and a Z, as well as having this particular variant, and it's got an AAT measurement, which is very much like someone who has a ZZ. So we can go and look at what are some of the kind of variants that have been found and that people have put on a website or have shared. And I'm going to go to this website here. Okay, and if we look on the left-hand side of the screen, we've got under clinical significance, we've got some variants that are conflicting. So one laboratory may have said this is going to cause AAT deficiency, and another lab might say, no, we have a patient that has normal distribution of their AAT. This button here, benign and likely benign, is that there's evidence that those variants cause no change to the AAT. We've got some variants where they remain uncertain, so it's not known whether they cause disease or they don't cause disease. And then we've got some clearly pathogenic ones, and by pathogenic we mean that there has been shown to be a clear effect on the AAT protein, and the AAT protein is limited or changed in some way. And then another set of variants which, to all intents and purposes, people believe they do change the AAT protein. So I'm going to select these pathogenic ones. And there are 50 of them.
Val, before, as you're doing that, we do have one question from CODIS saying, are the 50 tests a month for all of Australia?
No, so we very much serve, I suppose, the public hospital system within Queensland, perhaps a bit of northern New South Wales, that it's probably easier for some of the people who pathology providers to send the sample to Brisbane instead of sending it to Sydney. So yeah.
Very good. Thank you, Val. Thank you, CODIS.
Yeah. So as I've brought up some of the variants, and I don't know if some people are online and they might know what their variants are. It may be the Z variant that you have. You may have 2 copies of it, or you may have an S variant and you have 1 copy of the S and 1 copy of the Z, or you may have one of these rarer variants. And as I say, in this site here, it's got really the top 50, and it's got some information about what it affects, something about the clinical significance, and there is then some evidence about did one laboratory submit something or have multiple laboratories submitted something. So if I go back to my slides, I've gone to ClinVar, and then if I go page down, I didn't really want to go into too much more detail, but just to say, we rely upon the patients and your medical specialists sending samples to us for analysis. And of course, within the laboratory, there are a lot of different people who are involved in the analysis, from our central specimen reception, our DNA extraction, the staff that analyze them, and the various clinical staff that help in the interpretation.
Val, we have one more question here from Jody. She says, hello, I am PIZ type. What does that mean for me?
PIZ type. So is there one Z or more than one Z?
Her question just has one Z.
Yeah. So, the whole language of genetics is very complicated. So a lot of testing when it began was looking at the protein, and it was looking at the protein in the serum, and then people began to look at what were called gels or isoelectric focusing gels, and people were separating proteins on these gels based on size at a particular pH, which is whether it's an acid or a base. And they were usually measuring a band, and the band was moving between what was called a positive electrode and a negative electrode, and it took up a certain position. And then people started to call these by certain letters, and it depended on what position they were relative to each other. So the Z allele is the most common allele, and to call it a PI is really referring to the protein that has that Z variant. And then when we're in the DNA lab, we start talking about what is the nucleotide number or the C dot number, and then we can use that C dot number to work out what is the protein. So does that help to try explain things?
We'll see if we get a response from her.
Yeah.
In the meantime, I have a question also from John, and John is asking, are there indications of efficacy of AAT which are significant beyond levels?
I suppose I'm not entirely sure what John is asking there, but if you imagine when you've got a protein, and that protein is going to carry out a function, and the protein is very, I don't know, I always think when a cell has something, it carries out the different proteins carry out their jobs extremely well. And you could imagine that if you make a change, that protein is going to change in some way. Sometimes it will be change in a very subtle way, sometimes in a very severe way, but in a way you've got all the extremes in between. So from an evolutionary point of view, that protein has evolved to carry out every job it needs to do, say in the lung, in the liver, when it's in the blood, so that if you make a small change, and that happens at the DNA level, it's going to change the protein, and then it's going to change how that interacts, so that every change ends up being quite unique, or at least some of these rarer variants end up being quite unique and quite difficult to actually describe. So something like the S and Z, there are many patients, but one of the rarer variants, there may not be many patients about. And if you think about, the patient and the patient's family, is there someone else who's going to have the same genetic type of variant? There may or there may not be. And one of the things we can't do with human individuals is, make somebody else. We've only got who is there, and you can analyze them and then make comparisons between them. So I don't know whether I'm going off track.
Thank you, Val. Thank you, John, for your question, and thank you, Jody, for your question. Val, we have one that is going to come in here. Heather Lee has a question, and she wants to ask it verbally. So, Heather Lee, I'm going to unmute your microphone, and you are live. Go ahead and ask your question. And I don't hear anything. So, Heather Lee, if you don't have a microphone, go ahead and click on the Q&A button. And go ahead and type in your question.
Can you hear me now?
Yeah, there we go. Go ahead, Heather.
Okay.
Welcome.
Thank you. Good morning to all. My husband's recently been diagnosed with melanoma and is having a immune therapy. Will this Alpha-1 trypsin affect that therapy in any way?
Like for me, is it okay if I continue?
Yes, please go ahead.
Yeah. So for me, I'm very much a laboratory-based person, and really what you're asking is something quite specialized.
Oh,
and
okay.
I would say it's probably best to talk to your medical specialist and ask them. you know, if there is something that your medical specialist wants to know about the DNA side, so say he hasn't got a copy of a report, you know, then we can try and make sure the laboratory gets that report to him. And if there is some extra information he wants, to point him in the right direction, like for some of these websites that I've mentioned.
That would
So,
be good. Thank you very much. I will tell them about your websites and you.
yeah.
Very
And
good, Heather Lee. Thank you very much. Back to you, Val.
yeah, and if Steve has my contacts as well, so if you need to contact me directly, you can contact me through Steve, the president of the Australian or the Queensland AAT or AAA. So over to you, Steve, if you have anything.
Yeah, thanks, Val. So you're through your presentation?
Yep.
Sure. Well, I have a question or two that, if I could ask you, but maybe of interest to all others. Do you see an argument in Australia at the moment? The Alpha-1 testing is funded by the government for testing levels and for phenotyping via isoelectric focusing. Is there an argument for the government to fund DNA testing of Alpha-1, do you think?
Yeah, like you probably saw some of the numbers that I've given, and to some extent the pickup rate, you know, is actually quite low. Doing DNA testing is not really cheap. You know, to do the screen for the S and Z variants is in a way quite straightforward, and I would say it's a bit like hemochromatosis gene testing. So say from a Medicare point of view, it's not really very much money, but the DNA sequencing to find another variant is more expensive. And it's probably, you know, 5 to 10 times that price. But I would think, are the specialists and the genetic labs working together? So I think the genetic testing is a bit different to just sending a sample, getting a result, and reporting that back. So I think the genetic lab needs to talk to the doctor. I'm sure everybody knows that the AAT level changes, and it changes depending on whether the patient is well or may be unwell and may have various different infections or other medical problems. So you may take a measure of the AAT protein at one time, and then you measure it again and it's different. So I think we probably need better ways to focus on the individual that has a reduced AAT level, and then understand, is there a genetic cause for that? And so measuring the AAT protein just at one time and not doing it again may not be a good thing. And then, you know, a lab just sending the sample to us to do a DNA testing, and then we just send a report back, and in a way there isn't enough, I feel there isn't enough communication. And it's really how do we manage to build on that communication? And I suppose who is the specialist who will do that? And I'm not sure from the patient point of view and the society's point of view how people feel. You know, they're either their protein testing or their gene testing is done.
Yeah, I think collaboration, that sort of leads into another question. There
Yeah.
are a couple of others who have asked questions as well. Kevin, I'll hand over to you after this one, if I may.
Very good.
Yeah, just again, Val, some of the statistics that you presented in terms of phenotypes equating to various median levels and so on are quite interesting, but is there any, that's very laboratory specific.
Yeah.
The Medicare website has a range of statistics on how many tests are being done for the funded tests.
Yeah.
Is there any move towards, or is this something perhaps the association needs to drive, of compiling all of the statistics from the various laboratories so that we have like a national view, which is arguably more insightful given that it would be from a bigger sample?
Yeah, I think so. And like there is a study going on at the moment from the federal government, and the federal government is asking that question: what kind of genetic tests is each lab doing? Who are they doing that testing for? And it might be, what are the different specialities within a hospital or a medical practice? Is somewhere like Queensland, how many tests are we doing for what might be New South Wales or the Northern Territory? But, you know, I think what may happen is the federal government will do that, and they will do it through RCPA, which is the Royal College of Pathologists of Australasia, but they will be perhaps focused on maybe other genes, and they may be cancer genes or something like that. So I think you, as your society, wouldn't, should know what are the numbers for, your gene of interest. And one of the questions might be, if someone measures the AAT protein in serum, do they refer samples on for genetic testing? And what kind of genetic testing? So is it just a simple test, but say no variant is found? Does someone then say we need some additional testing? And the question, I think sometimes I'm asked is particularly around the null variants. So a null variant will make no protein, and it's more likely to have more effect on the patient's clinical picture. And I know that we do that extra gene testing, and there is another lab over in Western Australia doing that testing, but I'm not really sure whether there are any other labs.
Yeah, there's some interesting statistics, like with the two tests that the government funds.
Yeah.
There's lots of statistics based on gender and age at the Medicare website with the two tests independently. But I guess with those tests, it doesn't include actual results. But it'd be nice to bring those two tests together, because obviously phenotyping and levels is when you put that data together, it produces results.
Yeah, in our lab, we tend to measure this is Pathology Queensland, we tend to measure the level of AAT and then move straight to genetic testing. So not to do the protein studies from the isoelectric focusing point of view. So in a way, genetics and the sequencing of the gene gets in there to find exactly what the change is. And I don't know if you can remember the picture of that slide of the different parts of the gene. There were a hell of a lot of changes, different colors that were there. So there's a lot of variation out there.
Yeah, interestingly, this is period July 2017 through to January 2018, so a 7-month period. There were, across Australia, there were 12,721 tests done for testing levels.
Yeah.
And then 1,093, so what's that, about one in 6 moved to phenotyping.
Yeah.
So
And
there were
do you have any measure for the gene testing?
No, because it's not government funded, and therefore but whether the labs were doing the gene testing and then processing them as a claim under phenotyping, that could be one.
And to some extent, you know, I suppose being a geneticist or a molecular geneticist, I would say 12,000 people are tested, and then, you know, about 10% go on to do one other test. Should there then be another 10% of those which go on for a full gene sequence? And, you know, I suppose my feeling is perhaps there is under testing, but it's no good just getting a sample that comes in, you know, with a request form, and then you're waiting to pass it down, you know, a chain of analysis. If there is no communication between the various scientific staff and medical staff. So I suppose in today's consumer orientated society, we don't spend enough time saying, I haven't got the right answer, I need something more. And in genetics, we often talk about the diagnostic odyssey for a patient. So a patient may have so much testing done, but it never gets to the bottom of solving the problem.
There's a lot of work needs to be done in that area.
Yeah,
Kevin, I'll throw it over to you to move on to those other questions.
Very good. Thanks, Steve. So Val, we have about 4 more questions here. And it looks like the first one is from John. Back to John. He says, one issue that seems to determine effect of the ZZ phenotype is so-called cofactors, that is, other proteins that might have similar roles to AAT and can step into the roles. In my family, we have 4 ZZ siblings, 1 very affected, 3 not so much. So his question then is testing of related proteins relatively straightforward?
And I would say perhaps a simple answer is no, it's not. But, you know, I think, so say you have that situation where there are 4 individuals, they all have the same, what we would call genotype, ZZ, but there is different clinical profiles for each. You know, it's perhaps not unusual, but to get to the basis of that is probably not easy. And you could imagine that in that family there are 4 individuals, and perhaps it's quite an important resource. But how would you do that? Someone is going to have to say what the differences are and ask the questions. Is there some simple reason that would explain it? But say it is purely genetic, then we're really talking about trying to discover something new, which is not impossible, but not simple. And you know, when we talk about genetics, we always say that the phenotype, which is what the person has, is made up of genetics plus the environment. Now, you would think about a family and you would say the environment is likely to be very similar, but then there's the genetics, and is it 1 gene or is it more than 1 gene? So, yeah. But I think, you know, it's a valuable piece of information that needs to be captured in some way. And when we talk about the genetic databases that are there, and I showed you the ClinVar, I believe we need to be capturing more of that specific patient excuse me, clinical data.
Very good. Thank you. Thank you.
Kind of
Sorry to interrupt there. Thank you, John. Let's see, Val, we've got, next one is from, looks like Jody. Jody was the person with the PIZ type, and she wanted to know what that means for me. Apparently she had some computer issues at the time and said, sorry, I lost part of the answer, but I was told he asked if only one parent had the Z gene. Both of mine have it. I was wondering if that changes his response.
Yeah, and I'm sorry about the technical side of things. I know I'm quite technical, and this Zoom was a challenge for me. But if both your parents have a Z, and we would call them heterozygous, and then I would assume you have ZZ, then I would say that is the most common AAT deficiency genotype.
Okay.
Yeah.
And let's see, Codis has a question. Thank you, Jody. Codis has a question, says, how often do you think ZZ patients have their antitrypsin levels measured annually? I had mine tested twice in 5 years, and it has remained the same, 0.01.
Yeah. Like you probably noticed the values that I presented, and the way they're presented in Pathology Queensland are grams per litre. But it seems perhaps the norm is to measure milligrams per deciliter, which is milligrams per 10 mils. So it's probably making those comparisons are difficult. But, you know, I think the levels are changing perhaps when other infections occur. And my understanding of the general, and if I just call it the AAT gene, is that if there are infections, normally more AAT protein can be made. But one of the things with the ZZ is there is a protein made, but you can't upregulate it. So you can't make more of it when it's needed. And if you see the slides that I presented, and I think one reason I was doing it was just trying to see what were the AAT levels, and what was the mean value, and how did it change over time. And a lot of the values were very similar, and I wrote down some of the ranges. It was from 0.19 to 0.32. So, and I think, you know, one way the society might work is perhaps to gather some of this information, and maybe then be able to say, here is what happens, or here is what happens with our group of patients who are a member of our group. And I think when we talk about pathology, we talk about quality and improvements. And if someone can say, you know, at such and such a laboratory or in a certain state, here is what happens, but in another state, this is not happening. So could the society, you know, drive improvements and drive what would be called best practice? And, you know, there's a lot of information on the internet, but how do you bring it back to, you know, what is happening locally? And how do you find champions for your cause? And I was certainly, when I was trying to set up today, I was going to have a look at the Australian Alpha-1 Association of Australia, and I saw Steve's name, but then there was another position and it was unfilled. So, you know, who should you have that's part of your society that's helping you? And, you know, from my point of view, from the genetics and the genetic testing point of view, I'd certainly be happy to work with you, Steve, to, you know, meet up, whatever it is, on a monthly or 3 monthly basis and try and talk about some of these issues and try, you know, ask some of the questions that you then may be able to use to drive improvements for your group.
Now that's, I'm not sure if you can hear me, Val. Can you hear me?
Yeah, we got you.
Oh, okay. Yeah, no, that's terrific, and I might take you up on that kind of. Were there any other questions, Kevin?
Yeah, I have 2 more. And speaking of the association, Jenny is wanting to know, Val, if there's any connection between hemochromatosis and Alpha-1, as a few of our members have both.
I would say, you know, simple answer is no. And, you know, if you imagine your family and even your extended family, and I would know mine, it's not unusual to have, you know, 2 genetic disorders. And, you know, I think if you, you know, the more you talk to related family members, the more likely you are to find something. So something like hemochromatosis is in a way so common that it wouldn't be unusual to find someone with hemochromatosis and something else. And, you know, from an AAT point of view, I suppose it's not as common as hemochromatosis, but it's still not uncommon.
Very good. Thank you, Val. Steve, that's it. I'm going to bring your video live now. So you can go ahead and close this out as soon as you accept. There you go. There it is. There we go.
Terrific. No, that was most informative, Val. The whole webinar will be, I think, available via the Facebook website for watching by those who haven't been able to make it. But your time is really valuable and appreciated. And also we'll take you up on maybe working together to see if we can do something more with this national testing and getting some meaningful information from that.
Sure.
Thank you so much.
Okay, my pleasure.
Thank you, everyone. Have a great evening.
Oh, thank you so much, Dr. Hyland. That was great.
I'm delighted to be able to talk to you.
Have a good day. Bye-bye, y'all.
Thank you, Kevin.
You're welcome.